Hi, and we're joined by Trevor Peters, who's the CEO of Willow Biosciences. Trevor, how's it going?
Very good, thanks. Thanks for having me.
Yeah, no problem. So just looking at the past couple press releases, Willow's relatively new to the public markets. It looks like you started trading about a month ago.
Yeah, correct. Yeah, we closed a $29 million financing April 12th and began trading April 15th.
Great. So this being your first time on the show, why don't you give us a bit of an overview about what the company is all about and what you've been doing in the last little while?
Right. So Willow Biosciences is a synthetic biology company that for the past several years has done a lot of work in opioid research and ultimately commercializing or developing a commercial pathway for thebaine, which is a precursor molecule for all the semi-synthetics like oxy, hydro, buprenorphine, naltrexone, naloxone. And so we finished a program with our partner last year and then began working on cannabinoids about 10, 12 months ago, developing the same concept, which is a synthetic biology platform to produce what we refer to as an API or active pharmaceutical ingredient. So we look at the cannabinoids as an ingredient as opposed to a cultivation process.
Okay, but having said that, I guess you're trying to ultimately grow molecules specific to the cannabis plant.
Right. So when you think about the plant, and it has a variety of uses, some of which really only target the small molecules that the plant produces. And so when you think about cultivation for that purpose, you're essentially throwing away 99% of the plant to harvest the trichomes and the small molecules off the buds of the plant. And so what we view as a relatively inefficient process and costly, and something that we can accomplish via synthetic biology, essentially using yeast as a platform to grow the exact same molecule and do it in a much shorter time frame, 10 days versus a crop that would take 3 to 4 months to grow, and with a much lower cost structure based on our synthetic biology process.
So when you're growing the molecule, and you mentioned comparisons to the plant, like what type of scale, how can we imagine and sort of picture what it looks like?
So fermentation, and this is no different than, it's baker's yeast essentially that we use. It's no different than brewing beer with slightly more complicated, much more complicated science that's involved in actually developing the yeast strain. But that yeast strain, if you can imagine, 100,000 liter vat, for instance, and us producing at, to use a round number, 3 grams per liter of product, we can essentially in a 10-day period produce 300 kilograms of pure cannabidiol out the back end. So I'm not sure what the biomass equivalence would be in a greenhouse, but I'm sure you'd be talking about hundreds of thousands of square feet to come up with the same product over a 3 to 4 month period.
Wow. So when it comes to cannabidiol, and we know there's over 100 different cannabinoids out there. So if somebody's growing CBD, what does that mean in terms of, like, you can relate it to strains of plants that produce different types of CBD, I guess is the easiest way to understand it.
So the plant produces a lot of cannabinoids, as you mentioned, 100 plus or minus cannabinoids that come out of the plant. One of the challenges with that is not all of those cannabinoids are desirable. So for instance, THC, which is psychotropic, oftentimes you don't want that mixed in with CBD. For instance, Epidiolex, which is produced by GW Pharma, is used to treat children with epilepsy. Obviously, we don't want to be giving them THC. So our process is different in that we produce the pure isolate. So whichever cannabinoid we're targeting, CBD is the one I've mentioned, but any one of those 100 could form part of our process. And that's the nice thing about what we do, is for the end consumer, whether it's an industrial consumer packaged goods product manufacturer or a pharmaceutical company, we can really tailor that end product to what they have. So they've got repeatability, scalability, purity, and ultimately a very low cost solution.
Okay, so at this point in the business, you want to be able to grow kind of what you say you're going to be able to grow, or cultivate, or I don't even know what the right word is, but biosynthetically produce.
Yeah,
and then sell that to a large pharmaceutical company or whatever end application is, so you can provide the molecule they need, for cheaper and much more of it compared to glowing print.
Right. Yeah, cultivation. Yeah, again, we view cannabinoids as an ingredient, so we would be an ingredients manufacturer.
Right.
This is a bit different than, say, food or beverage in that we're producing a regulated product that ultimately needs to be high purity, so what we call API or active pharmaceutical grade, and our process tailors very nicely into that. I mean, biosynthesis in yeast in particular and other processes is used for producing insulin, artemisinin. There are numerous drugs, for example, that are in production today because of biosynthesis.
So who would your ideal customers be, and what kind of money could we potentially imagine that could be made from this?
Well, cannabidiol right now, I'll just touch on that last point. Cannabidiol right now in isolate form sells for anywhere between 10, 15,000 per kilogram. If you look at the plant-based manufacturing side, you're probably just under $10,000 in cost, all in. Cannabidiol, other cannabinoids are hard to isolate, purify away from the rest of the products in the plant.
Sure.
So you don't have contaminants. So it's fairly costly to manufacture and purify out your cannabinoid, which is why you see those costs. Our process is much cheaper than that in terms of our cost base. So we can produce well below, probably 90% less than what the cultivation costs would be. So you could imagine margins for us in that range. Ultimately, once we get up and running, we're still obviously in the research and development stage, but once we're up and running in roughly 2 years time frame, and we'll be producing essentially multi-tons of the product. So again, going back to my usage earlier of the 100,000 liter tank, if we're producing 3 grams per liter, every 10 days we'll be producing 300 kilograms of cannabidiol, for instance, or other cannabinoids that are desirable in the market. So you can do some math around 300 kilograms every 10 days, what we can do on an annual basis in that 100,000 liter tank, which we can do anywhere globally, and then the revenue of, say, $10,000 a kilogram plus in terms of what we could command for a price for that product. Again, that's an isolate. That's not a full spectrum extract or whatever comes from the plant. This is a very specific market. Again, CPG, pharma type customers who'd be requiring a highly pure product, which is why the cost base is what it is.
Yeah. Okay. So now that we kind of understand what you're doing and what the goal is, on the capital market side, what's the share structure like in the company right now?
So we just yesterday announced a 25-to-1 share rollback, which will ultimately give us, I think, 68.8 million basic shares outstanding.
Okay.
That starts. We start trading post-consolidation Tuesday of next week. So 68.8 million shares outstanding. We've raised the 29 million, as I said earlier. That leaves us in a very well capitalized position. We actually finished 2020 with just under $8 million in cash. That's about the time when we'll expect to start seeing, if not revenue, at least sales contracts that will take us into revenue in that time frame. So leaves us in a very good financial position, a healthy financial position. One of our major shareholders, Tuatara, a large U.S. investment firm, very focused in cannabis, has put $12 million into the company. They have also essentially an option to participate for another $12 million, which we expect them to exercise in that intervening 2-year period. So that'll be in addition to that $8 million that we finished 2020 with.
Okay, and management ownership of shares is
significant.
Yeah.
We've also participated, if you look at our slide deck, we participated for about $6 million of that financing, the 29 that we raised. We've also put in money prior to that. So management and insiders, I think we're on the order of 30-35% ownership in the company. So.
Great. So, and I noticed out of the 29 million, one of the major things you're doing is building out 3 labs: San Francisco, Vancouver, and Calgary.
Yep.
Is
so
that right?
we have existing labs now in Vancouver and Calgary. We're augmenting those and adding some new components to them, and that's an important part of what we do on the Canadian side. On the U.S. side, San Francisco is our 3rd site, and that's what we do. It's called high throughput mutagenesis and strain analytics. Essentially, turbocharging the yeast strain that we develop in Calgary. Pardon me. And that's where we are probably going to be spending a bulk of our capital. The high throughput, the robotics, the computational biology equipment that we need down there forms a big cost for us. San Francisco, we like to do it all in Calgary, but San Francisco is really where the hotbed of biotech and our staffing requirements really come from that environment. So, and then G&A and scale up will form part of that cost as well.
Okay, so the difference between Vancouver and Calgary, are they doing different things, or is it just sort of?
They're to you and I, they would be very similar,
right.
But to the scientists, no, they're very distinct. There's more of what I refer to as the macro understanding of the plant that happens in it's UBC, Vancouver, and then University of Calgary is where our Calgary lab is based. Calgary is where we do. Calgary is really the hub for us. That's where we do a lot of what we call discovery biology. So understanding the plant's metabolic pathway, the factory by which the plant actually produces cannabinoids, that occurs in Calgary. Our chief science officer is a tenured prof at the University of Calgary, a Canada Research Chair in plant metabolic pathways, and one of very few, probably 5 researchers in the world who can do the type of work that he's doing, understanding the plant and how to take that factory, the assembly line, transport it into a yeast cell and accomplish the same thing.
Great. Okay, and then finally, just quickly, when could we expect the augmentation process to be complete with the money that you raised?
Well, there's a number of components to it, and the main business line that I've mentioned to you, producing cannabidiol. It's something I talk about because people understand it, they're aware of it.
Yep.
We have a host of other ancillary business lines which we're pursuing at the same time because we are capitalized to do it. We think that over the next 12 months we'll be in a position where we can start to scale our cannabidiol process. And why that's important is as we go through the discovery biology phase, ultimately through the high throughput mutagenesis strain analytics stage, we will end up at a point where we move from the shaker flask, the test tube, to ultimately a fermentation environment where we're working in multi-liter tanks, and ultimately 10,000 plus liter tanks to scale up our process. So towards beginning of next year, we expect to be in that process, and that's sort of the halfway point to when we ultimately expect to be working in the much larger fermentation tanks and producing high enough concentrations, call it 3 to 5 grams per liter of product, where we can be doing. So 24 months from today, we'll be in the process where we can ultimately start working on sales contracts and ultimately distributing our product.
Great. Well, Trevor, thanks for coming on, and that's a great introduction to the company, and we'll have you back when your next milestone comes around.
Okay, great. Thanks very much, Fraser.
Thank you.
Appreciate it.